linked populations using new technology and advanced technology and methodology. But this afternoon's session will be a little bit different than this morning. Instead of taking a broad view of the technologies themselves, we've asked individuals who are working on particular species to talk a little bit about how they've used tracking technology and other technologies to establish migratory connectivity and link populations with their species. And the idea is then to have all participants sort of explore these particular studies as case studies. And after the individual presentations, we'll move into a panel discussion where it's my hope that those folks who are working on particular species can ask questions of the panelists from this morning and each other in terms of, what their best next step might be, or to share some of the more technical aspects or barriers, roadblocks, things that they overcame, and that might be helpful to other people who are beginning studies or are in the middle stage of studies on individual species. So it's intended to be a little more practical than this morning's session was. Once again, chat will be disabled until the conclusion of the webinar today, and then we'll keep it open for a half an hour for folks to do whatever they want in the chat and say whatever they want. But it's in the Q&A tool is always open, and you're encouraged to ask questions in the Q&A tool, either to individual panelists or to the group as a whole when we move into a panel discussion. You can maximize your view when you're watching as a participant by going up to the top of your screen and doing view options and setting up a side-by-side view where you can slide between the PowerPoint size and the participant video size. So without further ado, then I'm going to start us off. Our first talk this afternoon will be, in putting it into practice, will be from Ellie Knight. Ellie is a PhD candidate at the University of Alberta. She's studying the conservation of common nighthawks across the full annual cycle, and that will be the subject of her talk. Welcome, Ellie.
Sorry, Tom, just had to unmute. Can you hear me now?
Yeah, we're looking at your the view that you use to see your notes as opposed to your other view.
Okay, sorry. We practiced it too.
Wait. In the upper left of your screen, do it again. The upper left of your screen, it says swap displays.
There we go.
Oh, there you go.
Are we good?
Yep.
We're good.
Same thing. Okay. Thank you, Tom. Thank you so much for inviting me to share our work on common nighthawks with everyone today. We've been working on this for a while. And thanks also to Stu for the model species nomination. I think, well, I'll show you what we've got so far and then we can decide. So today I'm going to talk about how we've approached estimating migratory connectivity for the common nighthawk. This work is currently in its third and what I hope is final round of reviews at ecography. And so we've approached it with the idea that estimating connectivity continuously and comprehensively can be a first pass at narrowing down potential mechanisms of differential population trends. And the results can then streamline the process of quantitatively evaluating those potential mechanisms, which is going to be covered tomorrow in the linking vital rates session. So today I'm going to talk about the ins and outs of estimating migratory connectivity, and then we'll summarize the approach we've taken, and I'll run you through how we've applied it to this handsome creature here, the common nighthawk. So I put the acknowledgements up at the front just because I want to highlight how much of a collaboration this work really is. I'm deeply grateful to Pete and Aaron and Steve for their guidance and trust in me on this project, to Autumn, Lynn, Amy, and Janet for their wisdom and support, and to the many, many collaborators that have contributed in so many ways. Of course, to the funders that have made everything possible, particularly ConocoPhillips and Environment and Climate Change Canada. So Keith Hobson, I think, coined this sort of phrase in one of his papers. He coined migratory connectivity is a useful first step in evaluating causes of differential population trends among locations. And so today we're going to talk about how to take that first step. As Autumn Lynn mentioned, connectivity can be used to understand the factors that cause differential population trends, because when populations have high migratory connectivity, it means that individuals from each population stay together at other places in the annual cycle, and thus are vulnerable to local environmental conditions at those places. On the other hand, if connectivity is low, say between the breeding and wintering grounds, like we see in Will Cresswell's figure here, then individuals from all populations are subject to the same broad set of environmental conditions on the wintering grounds. And so we can conclude that the breeding ground component of overall population trend must be responsible for the overall differences in trend between populations. Now, migratory connectivity is often described as high or low, or Autumn Lynn said strong versus weak. But those are qualitative descriptors that do have the potential to be subjective. So instead, we can quantify migratory connectivity, which is typically done relative to populations on the breeding grounds. And there's a couple different approaches to quantifying migratory connectivity. The one I'm using today is based on the Mantell test, which is essentially a correlation of two distance matrices. But regardless of approach, they all provide an estimate of migratory connectivity between -1 and 1. And so in our example from Emily Cohen's paper on estimating migratory connectivity here, we see MC is approximately 1 when individuals that are close together on the breeding grounds stay close together on the wintering grounds. In other words, the populations are connected between seasons. If individuals from the breeding populations are highly mixed on the wintering grounds, we see little pattern, and so migratory connectivity is approximately 0. And of course, it can also be negative, which occurs when individuals that are close together in the breeding grounds are further apart on the wintering grounds. When using migratory connectivity to begin to understand the causes of differential population trends, it's important to think about your geographic extent, because obviously the value of connectivity will depend on the populations you include. So often connectivity can differ greatly between regional and range-wide perspectives. And so in this example, again, from Keith Hobson's barn swallow work, you can see that if we look at just the eastern populations, so that's New Brunswick in black and Ontario in red, that we see high mixing of individuals on the wintering grounds and less low migratory connectivity. But if we include populations from Western North America, we actually see a migratory divide that keeps Eastern and Western populations separated on the wintering grounds and results in high migratory connectivity. And so taking a range-wide approach can be particularly important if we're using migratory connectivity as a first step towards directing conservation efforts. But I would argue one of the most important components of using migratory connectivity to guide next steps is to evaluate it continuously across the annual cycle. So up until now, connectivity has only been evaluated between stationary stages. Most common is between the breeding and wintering grounds, like we see here in Gunnar Kramer's golden winged warbler work. But other authors have quantified it between the breeding grounds and major stopovers. But the challenging thing about conserving migratory species, as we all know, is that they occupy many places throughout their annual cycle, especially during migration when birds are on the move. And so in order to narrow down potential mechanisms of decline, we need to consider all places of the annual cycle where threats may occur, including during migration. In other words, we need to evaluate connectivity continuously. And to do that, we have to consider that connectivity can arise through separation in space or separation in time. So the type that we're most familiar with is spatial connectivity. In this example, we have 3 different populations of a hypothetical migratory bird that breeds in the Great Lakes area and migrates south to a stopover on the Gulf Coast before continuing on. If those 3 populations each use a different route and area for stopover, we have high migratory connectivity regardless of when each population migrates. But if our 3 example populations use a common stopover area, we can still get high migratory connectivity if they have population-specific migration timing, because those differences in timing still cause spatial clustering of individuals. And so the only time we actually see low migratory connectivity during migration is when species are using a common stopover area or route, and they have at the same migration timing. And when studying differential population trends, it's important to distinguish whether migratory connectivity arises through separation in space or time, because different environmental conditions can influence populations depending on which type of connectivity it is. So when migratory connectivity is spatial, populations can be influenced by conditions that vary in space. So in our example here, different population trends could be caused by differing levels of foraging ground habitat loss between the 3 population-specific stopover areas. When migratory connectivity is temporal, populations can be influenced by conditions that vary in time. So they may be using the same stopover area, but because they're moving through it at different times, they're still subject to pressures that vary temporally, like inclement weather or pesticide application. And so the approach that we took, we've coined connectivity profiles. In a nutshell, you estimate migratory connectivity at equal intervals across migration. And the profile itself is a smooth prediction of those connectivity estimates. And so in our simulated example here, we've built a spatial connectivity profile by estimating migratory connectivity at every latitude of migration. And you can see we've simulated a peak at a stopover at around 30 degrees latitude, and that's showing up in our profile here. And then we built a temporal connectivity profile by estimating migratory connectivity for every day of migration. And again, this profile reveals the simulated peak that we've put in around day 60. And so to build a connectivity profile, you either need an estimated migration route, which you can build from movement modeling data, even with sparse and sporadic tracking data, which I'll show you in our Nighthawk example, or you can use raw data itself if you have high enough resolution tracking data. So let's look at what connectivity profiles can tell us for the common nighthawk. So for those of you that don't know the species, it's a member of the nighthawk family. It's an aerial insectivore and it's a crepuscular species. It's a long distance migrant wintering the Amazon and Cerrado regions of South America. It has one of the largest breeding ranges in North America. And across that breeding range, there is variation in population trend, like we saw on the map that Autumn Lynn showed this morning. But overall, this species has a mean decline of 1.75% per year over the last 40 years. Unfortunately, we don't know very much about common nighthawks, likely because they're nocturnal or crepuscular. And so we're kind of at the bottom of that recovery period for this species. There's a myriad of potential mechanisms that have been proposed for their decline. And so because we knew nothing about connectivity going into the species, we took that range-wide approach. We selected 13 locations across the breeding range, and we deployed Argos GPS tags on adult males and females. And so just a quick description of these tags, because you'll see them again in subsequent talks. They collect high precision GPS points, but they then transmit them to the Argos satellite system after every third point collected, which is great because you don't have to recapture your bird to get the data. Which is super important for nighthawks. It does mean, however, that these tags are really pushing the bounds of technology, and they're not perfect. So of the 85 tags that we deployed, we heard from 54 of those, and the distribution of data across those tags was sparse and sporadic. So between 30 to 60 points per tag, sometimes from different times in the annual cycle even. And so because we had sparse and sporadic sampling, that's why we used movement modeling to get a mean predicted migration path for each individual. And here's what that looks like for fall migration. So when we run connectivity profiles on these migration routes, we see that because this species is essentially using a common fall migration route after convening along the Mississippi Flyway, that there's low spatial connectivity outside of North America during fall migration. High interpopulation variation and fall migration timing also means that temporal connectivity quickly decreases to near zero and stays there. And so I should mention, although it does look like there might be some bumps that could be peaks in these connectivity profiles, none of them are significant. So we developed a two-step approach to evaluating each peak, which I won't get into here because it's pretty complicated, but none of these bumps made the cut. And so over winter, we did evaluate relative to the stationary wintering stage. And so we do see quite a bit of mixing. Migratory connectivity between the breeding and stationary wintering grounds was 0.29 in early winter, and then dropped down to 0.2 as several individuals actually moved to secondary locations. And so this type of sort of low connectivity or weak connectivity is common in long distance migrants. Moving on to spring, we see that spatial connectivity stays low for longer because birds take a more direct route back to their breeding grounds and so remain mixed for more of the spring migration. We do, however, see a pretty distinct peak in spatial connectivity before birds cross the Gulf of Mexico. And this peak does happen to correspond with one of the major important spring stopover locations that Nick Bailey and his co-authors identified in their 2017 review. And so the other peaks in this profile were evaluated as false or non-significant. And then unfortunately, temporal connectivity doesn't tell us a whole lot. There are a couple of false peaks in this profile, and then we're seeing really high uncertainty early on during migration. So let's bring our results back to potential drivers of differential trends and look at times in the annual cycle when populations have elevated migratory connectivity and thus are susceptible to local environmental conditions. So based on our continuous analysis of migratory connectivity, we could see those drivers occurring during fall migration in North America when connectivity is still high. But they're unlikely on the wintering grounds because we saw relatively low migratory connectivity there. We could see those drivers occur at the area before Gulf crossing during spring migration, but perhaps not during other times or places of spring migration. And of course, those drivers could still be on the breeding grounds. They could also be due to interactions between any of the seasons, like connectivity and habitat selection, which Calandra is going to talk about a little bit later this afternoon. And so this allows us to start narrowing our lengthy list of potential mechanisms. So here's an example of a couple of them in the seasons during which they might occur. When we apply what we've learned about connectivity, we can actually eliminate one and start to actually attribute locations to some of the rest of them. And so that's great. We're starting to narrow our focus for next steps. But what are those steps? There's still lots of potential mechanisms. How do we figure out which ones are contributing to declines? Well, I'm really excited to see what folks like Clark Rushing have to say tomorrow in the Vital Rates session, because that's the next step for this project. So not only did we plan our deployment locations to span the breeding range, but we also planned them to span a gradient of natural population trend from steep declines like Arizona to the one increasing population in Oregon. And so you can see how this sort of full annual cycle perspective is really allowing us to start strategically climbing that pyramid towards recovery and following the road to recovery for this species. And so with that, I'm just going to end by thanking all of the folks who have helped out with this project. It's certainly no small feat, and I'm deeply grateful to each and every one of them.
Thanks, Ellie. I think we have actually a time for a question. So Anna, do you have a question from the Q&A that someone might ask?
There are no questions in the Q&A yet. I have a question though, if that's okay. So how do you think you would incorporate sort of the strength of migratory connectivity into an IPM or some other full annual cycle population model? So if you have vital rates from certain populations on the breeding and non-breeding grounds, and you know that there's sort of 0.3 or 0.4% migratory connectivity, how would you incorporate that into a population model?
Yeah, so that's a really good question. And it's something that I've just started thinking about. I mean, so we have this ability to quantify migratory connectivity, but what do those numbers actually mean? I don't think anyone's tackled that one yet. I think, I mean, right now I'm sort of heading towards the approach that Clark has taken, where it's more relating the environmental conditions from across the annual cycle directly back to those vital rates, as opposed to integrating the migratory connectivity values into an IPM. But I think that that's an interesting avenue to go next. Is, you know, I think there's a strong potential for some simulation work on sort of, are there thresholds in the value of migratory connectivity that actually relate back to the vital rates?
Mm-hmm. Thanks. Tom, is there a time for one other question?
Come up, yeah, let's do it.
Okay. So there's a question in the Q&A. Do young birds follow the same migratory pathway as adults and or disperse to other populations?
Mm-hmm. That's a great question. So, I mean, this comes back to how little we know about this species at this point. The work that I've done has not tracked any juvenile birds to date. Alora Graham, who is in Ryan Norris's lab at the University of Guelph, is doing some MODIS work with juveniles and adults, and I don't know what her results are on that. I can tell you that we have had some birds that look to be doing some dispersal sort of from subadult to adult, and that dispersal distance is quite far in the sort of hundreds of kilometers range. But yeah, I think the dispersal question is a really big one for this species, and hopefully we'll get the chance to do some more work on that in the near future.
Thanks, Ellie. That was That was really good. Really interesting. Thanks for picking up. Thanks for thinking ahead and hearing Stu's comment about Nighthawk 3 years ago when you started work on this. Our next speaker is Calandra Stanley, and Calandra is a postdoctoral researcher at Georgetown, and she is interested in novel tracking technologies to reveal the movements of migratory birds across multiple scales and to try and understand the drivers and the consequences of those behaviors. And you're going to be talking about wood thrush, Cassandra.
Hi, yeah, thanks for the introduction, Tom. Hopefully you guys are seeing the correct screen. Yep.
Yeah, we are.
Okay, perfect. I'm really happy to be talking about this project on building a connectivity map for wood thrush, which I've been working on for over 10 years now. And so the wood thrush has kind of been a poster child for the declines of forest songbirds. And I believe they're currently listed as a 3X species in the Road to Recovery list, but that may have changed. But overall, over the last 50 years, their global population has declined by 60%, according to Breeding Bird Survey data. And unlike some migratory species, we actually have a pretty good understanding, and we've known this for a long time, of the general distribution of wood thrush across the annual cycle. So they breed in the eastern United States and Canada, and they spend the winter in southern Mexico and Central America. However, like Ellie just described and we've been hearing today, in order to really accurately predict the factors limiting wood thrush populations, we really need to understand how populations across the breeding range are connected between different periods of the annual cycle. For smaller species like migratory songbirds, technological limitations have really hindered our ability to decipher these patterns of migratory connectivity. And it wasn't until the late 2000s that tracking technologies really became small enough to deploy on these species. And so really part of the reason we began developing this connectivity map for wood thrush was because it was one of the few migratory songbirds that could carry these new tracking technologies. So unlike the other presentations that we're going to hear this afternoon, the two devices we use to develop the connectivity map for wood thrush are archival devices. So unlike transmitting devices, the tags, we need to recover the tags the following years in order to obtain the location estimates. So the first devices that became available for small birds were archival light level geolocators. And these devices record ambient light level, and based on day length and time of sunrise, they can estimate locations twice per day. But as you can imagine, there's a large degree of error associated with these location estimates. But nowadays you can get archival light level geolocators that are as small as 0.3 grams. The next technology that became available for smaller species like wood thrush were GPS archival devices. And these tags allowed for really accurate location estimates up to 10-meter accuracy. However, they were limited in the amount of location estimates you could get each year. And so the model we used for wood thrush was the low-tech Pinpoint 50 model that allowed us to schedule up to 50 locations across the annual cycle. We didn't necessarily get 50 locations. And so these are the two technologies we used. And over the course of 7 years, we deployed archival light loggers on 7 breeding populations and 4 wintering populations. And then starting in 2014, we additionally deployed archival GPS tags on 4 new breeding sites and 1 repeat site. And so the goal was really to try and get an idea of range-wide connectivity between breeding and wintering locations, but also to assess connectivity at major stopover sites. And so really, just like Ellie's project, this has been a huge collaborative effort that's taken over a decade. And here I'm showing kind of 5 of the major organizations, but each location had additional organizations involved in the project and multiple funding sources in order to get all of this field work done. So first I'll focus on the breeding to winter connectivity that we looked at for wood thrush. So all the deployment locations are shown as diamonds. And we grouped our breeding deployments into 4 regions and our winter deployments into 3 different regions, which I've color coded to help to visualize the data. So first, let's look at what we got for light level geolocators. We were able to retrieve data from 102 individuals tracked with light level geolocators from winter deployments that went to the breeding ground and breeding deployments that went to the wintering ground. And so if we overlay those regions from the last slide, we kind of see this leapfrog migration pattern for eastern breeding populations, where the more northern eastern breeding populations winter further south in the wood thrush range, whereas the more southern populations winter further north. And the Midwestern populations are at the most northern end and the most eastern range of the wintering area. But overall, if you see, there's a lot of mixing in the colors, and so a lot of mixing of different populations that we see in this data. And I also want to note the error bars associated with these location estimates. So I only have them on a couple of locations here just to simplify the graph, but they really are associated with every point. And so this really shows how when you're using light level geolocators, you're really only able to provide kind of large core scale estimates of migratory connectivity. And so that kind of begged the question, is the population mixing that we are seeing based mostly from technological limitations, like the location error, or is it actually population mixing? And luckily, the archival GPS tags came available, which have habitat scale resolution. We deployed them on the breeding ground. And here I'm showing the breeding deployments as larger circles. And as you can see, even with these higher resolution tracking data, we're still seeing a lot of population mixing. And so we can quantify the degree of migratory connectivity, like Ellie discussed. And we took 2 approaches to calculating migratory connectivity. With light level geolocators, we developed a migratory network that estimated the proportion of the global wood thrush population and how it moved between different breeding regions and wintering regions. And what we found is that the network estimated that 56% of the global wood thrush population wintered in the eastern range of the wintering area. And these birds were mostly coming from the northeastern part of the wood thrush range, whereas 41% of the global population was wintering in the central part of the winter range. And these individuals were coming from central and southeastern and midwestern populations. And then similar to Ellie, we were also used a migratory connectivity index to calculate migratory connectivity. And I did this using the different tag types that we deployed. And across all the different tag types, we get the same conclusion that wood thrush show moderate connectivity between breeding and wintering locations, with numbers ranging from 0.3 to 0.4. So regardless of the tag type we use, we get similar estimates. But what about stopover locations? Well, one of the huge disadvantages to using light level geolocators is that you cannot estimate latitude around the equinox. And so you're really limited to describing migratory routes based on longitude estimates. However, archival GPS tags allow you to actually identify stopover locations. So that's what I'll focus on. And this graph here shows all the stopover locations we found from the GPS tags. Each point is a location, and the lines are just connecting different locations. The eastern birds are in warm colors and the western in cooler colors. And we focused on the northern Gulf Coast stopovers because that's where wood thrush spend the longest amount of time on stopover. When we look at fall stopover sites, we really see this that the eastern populations stay to the east of the Gulf Coast, whereas Midwestern populations are further west. And we see higher indices of migratory connectivity due to these population-specific routes. By contrast, when we look at spring stopover locations, the birds are shifted further west, but they're also clumping together a lot more. And so we get much lower estimates of migratory connectivity. And it also suggests that this particular region of the Gulf Coast is seeing basically the global population of wood thrush, at least that we tracked. And so is important kind of bottleneck. And so overall for wood thrush, we found that migratory connectivity varies across periods of the annual cycle. And how we can then use this information to inform full annual cycle population models for wood thrush will be discussed by Clark Rushing tomorrow. So when designing a new project to assess migratory connectivity, which archival device is best would really depend on your research questions. So the costs of GPS geolocators are much higher, but you get this habitat scale resolution of location data. But depending on what you're interested in, you might not need that resolution of data. And so for wood thrush, when we look at breeding to winter connectivity, we see that both types of tags gave us similar answers. However, if you're looking for stopover connectivity, really only GPS geolocators can give you that information. By contrast, if you're interested in temporal connectivity, like Ellie was talking about, or timing of migration, light level geolocators are something that are going to give you location estimates twice a day, which is way higher than GPS tags. However, we are going to get a sampling bias due to both technologies because they're archival. So we lose information about where individuals are dying, and it means that we're only getting information for individuals that survive and return to our study area. And it can also lead to preferentially deploying at sites or on individuals where we expect to have high return rates. And so all these ideas need to be weighed when choosing a technology for answering these questions of migratory connectivity. But I also wanted to quickly touch on other full annual cycle questions that we can begin investigating as the spatial resolution of tags improves. And so we can look at questions such as research into niche tracking across the annual cycle, or simply looking at the habitats occupied by the same individual or populations across the year, and how habitat selection or habitat requirements might vary across the annual cycle. And so we were able to do this with wood thrush using the data we collected from GPS tags. And what we found was that wood thrush shifted to a more generalist habitat selection during migratory periods compared to breeding or wintering periods. Kind of a simple way to visualize this is that we looked at the diversity of land cover types occupied by individuals during different seasons. So here, different colors represent different land cover types. Each bar down here is a different breeding population during the breeding season or winter season. And the y-axis is just the proportion of time individuals spent in each location, each land cover class. And we can see that during the breeding and wintering ground, birds are predominantly in forested land cover types or in landscapes that are a mix of agriculture and forested areas, which is what we would expect for a forest breeding species. But when we move into the migratory periods, we're seeing individuals occupy a lot more novel land cover types, suggesting that the factors driving habitat selection preferences might be changing across seasons, which could have important implications for the management of this species. So there's a lot of exciting questions that we can start asking as technologies continue to improve. And so really assessing migratory connectivity is this critical first step. And for next asking questions about full annual cycle models, which Clark Rushing is going to be talking about, like I mentioned, about wood thrush. We can also start identifying migratory bottlenecks like we saw during the spring migration in the Gulf of Mexico, and start asking more fine scale questions about seasonal changes in habitat use and niche tracking. And really the choice of technology is going to depend on your specific research interests and questions. And that is it for me. And I want to acknowledge some of my collaborators and supervisors and funding sources. And yeah, this project took a lot of field work to evolve, so a lot of wonderful volunteers and technicians.
Belandra, I think we have time for a couple questions. Thank you very much. I really appreciated your outline of the different devices and their sort of strengths and weaknesses based on the questions that you're asking as a researcher. So thank you for doing that.
Still no questions from the Q&A for this talk. Tom, do you have any you want to ask? I also have a question if you don't.
Go for it.
Okay. Yeah, I was really curious. Do you have any ideas, Callie, about why the northernmost breeding populations winter further south, and then the southern breeding populations tend to winter further north. So like they seem to have an overall like shorter migration.
So the idea of these leapfrog migrations is that and we've kind of shown this looking at NDVI values, but the more southern breeding populations actually seem to be potentially better quality habitats, so more resource rich. So they actually provide a higher quality habitat that might allow migrants to fuel these longer migrations compared to the more northern ranges, which just are typically drier in the Central America. And so that's one of the reasons people have suspected that you might see this leapfrog migration pattern.
That's really cool. Thank you.
Yeah, great question. Anna, are there any other questions waiting for us in the Q&A?
Yes, I'm looking at them now. Okay, Michael Reed has a question. Fantastic talk. The strong separation between populations in fall stopover makes me think about hurricane risk for fall migrants. Is there any signal in the population trends that suggest that might be a problem?
So you are seeing some, so for example, the eastern populations are basically going down to the tip of Florida and then hopscotching to Cuba and then into Central America. Whereas a lot of the Midwestern populations are actually crossing the Gulf, taking a full Gulf crossing. And so, yeah, potentially those Midwestern populations are taking a much riskier migration route. And so if a hurricane hit in the middle of one of those migration routes and they're in the middle of the Gulf Coast, they obviously aren't going to be able to do anything about it. So it would be really interesting to look at those questions about the choices being made between those different populations and the risks associated with them. And if we started getting satellite tags that were small enough to put on wood thrush, we could maybe start looking at where differences in mortality patterns across different migratory routes.
Great.
How are we for time, Tom? Another question or no?
Sure, if you have one.
Okay, yeah, there's a question from Sarah Kendrick. She says, are these results, so the importance of the Gulf to a particular percent of the population, shared with state land managers to help bring further relevancy to their habitat conservation efforts? This would really resonate with those focusing on land management. It makes it personal to their state and region and their work.
So, I mean, the results have been published, but in terms of actually sharing with state land managers, especially with these really large range-wide projects, no, I'm not particularly involved with individual state managers, but I do think it would be really valuable for some kind of mechanism to be in place to allow researchers to connect with these people. I think that is hugely valuable, especially as we see the Gulf region being a really critical migration region for wood thrush and all populations of wood thrush.
Great. Well, thanks, Kylie. We're going to move on now to the next talk, which is going to jump taxa and talk a little bit about shorebirds. Laura McDuffie will be talking to us about lesser yellowlegs. And Laura, you're communicating to us from Anchorage, Alaska, where you're currently a biologist for the U.S. Fish and Wildlife Service, correct?
Yes, correct. And I'm also a master's student at the University of Alaska, Anchorage.
Well, great. Can't wait to hear about lesser yellowlegs.
Cool. Well, thanks, Tom. I'm really excited to share some current work that's been done on lesser yellowlegs. And so before I begin, I'd just like to mention my co-authors, Katie Christie, Audrey Taylor, Autumn Lynn Harrison, and Jim Johnson. So I'm going to begin by discussing the status and threats of the species, as well as some goals and objectives of the study, talk about the tracking method used to kind of reach our objectives, and then an example of how we're putting this into practice, and then some thoughts on where we can go from here. So the lesser yellowlegs has experienced a pretty precipitous decline since the 1960s, since 1966 of over 90%. And so as Ken mentioned earlier this morning, as well as at the July Road to Recovery Phase 1 workshop, He introduced this species prioritization matrix that kind of builds on previous vulnerability scores. And it includes these 3 different metrics. So there's high vulnerability, meaning that the species is on a watch list. The species has experienced a steep decline, so its population has been reduced by at least 50%. And then there's this last matrix called urgency that was first introduced by Partners in Flight back in 2016. And you can also think of this as half-life. So it's basically how long into the future it'll take a population to lose an additional 50% of its population. And so for the lesser yellow-legs, this is pretty startling. So it's predicted that within just 11 years, this population or species population could be reduced by about 58%. And so some of the presumed threats that this species faces includes loss of habitat. So for example, agricultural expansion and intensification, which then can lead to the use of agrochemicals such as pesticides, fungicides, herbicides. And then there's climate change, of course. An example of this may be wetland drying in the boreal forest, where these birds breed. This could be due to permafrost melt, but also these increased temperatures in the subarctic could result in elevated evaporation of these wetlands. But shorebird harvest is an actual known threat for this species, and it is likely the most harvested shorebird species in the Caribbean and northeastern South America. And it's estimated that between 35,000 and 66,000 individuals are harvested annually just within Guadeloupe, Barbados, Martinique, and Suriname alone. And so harvest is regulated in many regions, including those that I just listed. But it is illegal in others, such as Guyana and Brazil. And so really it's even though there's regions where harvest is legal, the number of lesser yellow-legs may go unreported. So this cumulative unreported harvest really is what we're concerned about, because harvest may be exceeding this potential biological removal, which you can also think of as like the sustainable mortality limit. And just to mention, this picture here is actually a bird that was banded in Yellowknife in 2019, and it was harvested in Guadeloupe on August 15th of this year. So the ultimate goal of our study basically is to understand what is causing the decline of the lesser yellow-legs. And so here I'm going to highlight how we can use baseline information such as migratory connectivity and the understanding of different migratory routes to apply different conservation questions such as exposure to shorebird harvest. So there are 3 pretty defined exposure zones that I'm going to highlight here. So there's the Caribbean zone, which includes basically the Lesser Antilles. Then there's the coastal Guyana zone, which includes Guyana, Suriname, and French Guiana. And then there's this coastal Brazil zone, which includes the coastal portions of several northeastern states of Brazil. And so the main objectives were to identify the probability that a lesser yellow-legs population is occurring within a harvest exposure zone during autumn. And then second, that we wanted to identify the period when these populations have the highest probability of occurrence in autumn. And so when I'm mentioning populations here, I'm really referring to more like a natural population, which are groups of individuals not divided geographically rather than genetically. But then of course, I'm also only referring to birds that were tracked in this study. So the method used to meet our objectives was the use of these Argos GPS pinpoint tags. And so these were just mentioned by Ellie a couple talks ago, and they worked really well for this species. They collect accurate data up to 10-meter accuracy. They have flexible scheduling. So if you're interested in full annual migration, you could schedule them to collect data throughout the full annual cycle, or if you're interested, say, in just autumn, you could have them collect locations more frequently, although everything is dependent on battery life, and on average, they only collect about 90 locations. And they also do have these past prediction capabilities, so it allows for this remote transmission of data, so you don't have to actually recapture the individual to get the data back. And so tags are deployed at 6 different sites across their breeding and post-breeding range. This value, the sample size N here, indicates the total number of tags that were deployed in 2018, 2019, and 2020 combined. So our largest sample size is here in Anchorage, Alaska, followed by Churchill, Manitoba, Mingan, Quebec, Yellowknife, Northwest Territories, Canadian National Wildlife Refuge, and James Bay, Ontario. So here I'm going to show you a very quick animation. So these are all the individuals that we tracked in 2018, 2019, and 2020 combined. And it is quick, but you can see that there are some different routes among these different breeding and post-breeding populations. In autumn, we tracked 63 individuals. And then in spring, we tracked 27 individuals. And so as you can see here, again, it looks like the Anchorage birds are arriving back at their breeding sites slightly earlier than the Churchill birds. So that was really quick. So here's basically a static representation of that animation. And so each of these track lines is basically an average track of all those different individuals. So of those birds tracked during autumn migration, which again was 63 individuals, we did see different routes among these different populations. So the Alaska populations, they followed the central Mississippi flyways, whereas Yellowknife and Churchill really followed the Mississippi and Atlantic flyways, and then James Bay and Mingan pretty much followed the Atlantic flyway exclusively. And so we're seeing, if we look in this region where all this harvest is occurring, the migratory connectivity between their breeding sites and this harvest zone is pretty weak. So as a reminder, that means that there's a lot of mixing going on between these populations. So then here in spring, again, this is a smaller sample size, 27 individuals. We're actually seeing this really narrow corridor for the Alaska Yellowknife and Churchill populations that are using the central flyway, whereas James Bay is still kind of remaining in this eastern Atlantic flyway, but it has this little loop going on up here as well. So then to kind of apply migratory connectivity, we are looking at harvest exposure. So this figure here shows the probability of a randomly selected individual from each population occurring in any harvest exposure zone or outside the zone. And so this is exclusively a binomial model with random effective individual. So if a bird was inside a exposure zone, it's given a 1, outside 0. And so the migratory flyways appear to predispose the Eastern Canada populations to harvest. And this is because harvest occurs within that Western Atlantic flyway. So we're seeing that this James Bay and Mingan population really has a higher probability of occurrence than, say, Anchorage and Canudi, which is down here at basically 0. And we're also seeing a kind of temporal difference in this peak occurrence of exposure. And as you can see here, Churchill's peak really occurs before the James Bay and Mingan populations potentially even arrive in that in those exposure zones. And so this is likely a function of departure from breeding sites, as well as the timing of how birds actually move through the exposure zones and further down into South America. But understanding the timing within each zone really is key, because harvest does not occur at the same time in every location. And so risk to harvest really does fluctuate. So for example, in Barbados, the open season for shorebird harvest is between 15th of July and the 15th of October. So if a bird is present in that area or is not present in that area during that time, then the exposures would be much less, right? So the main takeaway is that the whole species is not experiencing the threat of harvest equally. And so understanding migratory connectivity and exposure harvest really is just one component of the larger picture of lesser yellow lakes conservation. And so like I mentioned before, not all populations are exposed to harvest equally, which means then we actually need to explore what other potential threats may be eliminating the species. So for example, we could look at pesticide exposure within primary stopover sites, such as the prairie potholes. That might be a limiting factor for this species. Or we could look at anthropogenic disturbance here. So habitat alteration to pasture lands or agriculture, or even for recreational opportunities for people. That really could be a limiting factor for this species as well. And additionally, we're currently looking at this kind of genetic exploration. So we are building a genoscape model. We've got a lot of feathers. And so we're trying to determine if these different breeding populations, post-breeding populations are genetically distinct from one another. And then when we have feather samples collected in these harvest areas, such as Martinique here, we can use those feathers to then link them back to their breeding origin. But then on top of all of this, there's breeding ecology, such as nest success or adult survival, and that can also really indicate how a species is doing. And so like Ken Rosenberg brought up during the panel discussion earlier, he's saying, oh, it'd be really great to have a model species that is in decline, that we can kind of use a combination of all these different technologies to answer conservation questions. Well, for the lesser yellowlegs, we've already really begun that. So it's pretty exciting. And so finally, collaboration really is essential for the success of conserving lesser yellowlegs. And although the study does involve many partners already, there are still significant gaps that could be filled through additional collaborations. So for example, the species, we know that it's distributed along the Pacific coast of the US, but during our tracking study, not a single individual occurred in that area. So it'd be kind of interesting to see, okay, so what are the breeding origins of those birds? We don't really know that. And then second, maybe if we want to use or understand the species use of regions in South America during the boreal summer. So with birds that are not breeding, how do they use different regions within South America? So I like to just thank all my funding sources and mention all the different collaborators that helped make this project what it is. And yeah, I'd just like to say thank you. And there's my contact information if anybody has any questions. Thank you.
Thanks, Laura. That was great. I think pointing us to what we'll begin to talk about tomorrow, especially where we're not just trying to figure out where and when, but also kind of trying to make those inferences about what, and then somehow stitch it into a big picture. So great job. I think we have time for a question or two. I'd like to open that up. Anna, do we have anything from the chat or the Q&A?
We do not yet, but based on the other talks, I think they'll come in pretty quickly.
Laura, so a question I have is, in the shorebird world, was the hunting issue identified prior to the understanding of migratory connectivity, or was it the migratory connectivity that led to the discovery of the hunting issue in that eastern part of the northern South America and the Caribbean?
Yeah, so harvest has been known for a very long time. So it was kind of initiated all by the Atlantic Flyway Shorebird Initiative many years ago. So harvest was known to occur, just didn't really understand how the species or these different breeding populations are affected by harvest. So yeah, this really was kind of like a novel study to be able to understand that a little bit better.
Okay, Pete says he has a question. So go for it, Pete.
That was a great talk. Thank you. I was wondering if you could talk a little bit about the attachment techniques you use with lesser yellowlegs. Shorebirds are complicated for a lot of reasons with attachments. And did you use just a chest loop harness or leg loop harness? Or if you could talk about that a little bit, that would be great.
Yeah, so going into this, didn't know if these tags were going to work at all for this species. So it was a little bit scary starting out my master's and not knowing what was going to happen. But we ended up using leg loop harnesses. So we use Stretch Magic, which is kind of it stretches a little bit. It's like a jewelry cord. And for each bird, we actually fitted the harness to the unique individual. So they weren't like pre-made harnesses that were a certain size. We actually fit them on the bird. So we used jewelry crimps to try to tighten them and loosen them to make sure that they fit correctly. But yeah, to answer your question, we did use leg loop harnesses exclusively.
All right, we have another question in the Q&A. Kristen DeBoer says, I noticed that you have many fewer birds returning in spring relative to the birds going south in the fall. Why do you think this is? And also, can you estimate adult survival using your data?
Yeah, so these tags do have a finite life. They're not solar powered. You charge them once, deploy them, and that's how long they last, basically. So for some individuals, we did have these tags collect locations more frequently in autumn. Therefore, we didn't have enough battery life in the spring to collect data as well. There's also the chance that several individuals did die, so that reduced our sample size as well. Yeah, what was the last part of that question?
The second part was, could you estimate adult survival using your data?
Right. I think we could. It's a little difficult. We also are just exclusively banding birds and not putting tags on them. So we are we've had several years of data right now, resighting individuals that return back to Anchorage at least. And most of them that come back are just exclusively banded birds. They aren't carrying the GPS tags.
You mean you're going back to those old methods of banding?
Yep.
That's great. Well, let's move on. Thank you so much. That was great. That was really interesting. Our next speaker is Juliet Lamb. And Juliet is a Marie Curie postdoctoral fellow at the Center for Functional and Evolutionary Ecology in, I guess, Montpellier, not Montpellier, Vermont, but Montpellier, France. So thanks for joining us, especially at this late hour for you. And
thank you.
And Julia, you're going to take us one step further back toward the base of the bird evolutionary tree by talking about waterfowl.
That's right. Okay. So the project I'm going to be talking about is work that I did with the University of Rhode Island. So there's a lot of collaborators that I'll mention at the end of this, but as all the previous presenters have said, these kinds of projects are incredibly collaborative, and a lot of people participated in collecting data for this project. So I'm going to be talking about a large-scale study of sea ducks, but before I get into the species, I wanted to talk a little bit about methods. So to kind of take a big step back, we're often interested in questions that, especially with tracking data, that require taking individual data and translating it into some sort of system-wide perspective. So it's helpful for me, I guess, to think of it in the kind of particle physics framework. So you've got Eulerian movement, which is you're a fixed observer and you're watching individuals move by, or particles move by. And you've got the Lagrangian perspective, which is what you get from tracking data. And that's an individual's perspective moving through the system. And when we're using tracking data, we're often trying to take this Lagrangian data and put it into some sort of Eulerian framework, because conservation is generally done in a place-specific framework rather than an individual-specific framework. But as you can see from this cartoon, there's differences in perception of what's happening in a system, depending on whether you're looking at it from a stationary perspective or from the perspective of a moving object. So I think it's worth keeping that in mind. And when you think about different data types and how to kind of synthesize them together, because all of them provide valuable information, but it often takes some manipulation to get them into the same conceptual framework. So I don't need to explain migratory connectivity to anyone, especially after all of the talks that you've been seeing all day, but you've all seen these kinds of diagrams. And migratory connectivity is essentially an approach to taking this Lagrangian data and putting it into some kind of Eulerian framework. So how are individuals connecting different points across the landscape? And then to scale up and actually use that information to understand flyway or population level patterns. And so when I look at a model like this, the immediate thing that comes into my head is that this looks like a network model. So what is a network model? It's basically, I don't want to get too into the mathematics, and you'll have speakers over the next few days that I think will get into some more technical detail about network methods, but basically graph theory is the mathematical analysis of relationships. So in a graph, the nodes are entities. They can be locations, individuals, genes, depending on what your question is. And edges are the relationships. So it's these pairwise connections. How are each node in the network connected or not connected to each other node in the network? So it's a very simple concept and a very simple basis, but you can actually get some very powerful information from it. So commonly network analyses are used to look at social structures. So this is probably the context in which you've seen network analyses used the most. This would be a network where each individual is a node and the edges are some kind of relationship between nodes. So this example is from LinkedIn. It's looking at who's friends with whom, and you can already see that there are these sub-networks that start to pop out, which are the colors in this graph, depending on which group of individuals is more connected to one another versus their connections to the broader network as a whole. Another use, and one that's more similar to migration questions, is routing problems. So when you're using a GPS, the GPS is deciding based on network analysis which road you should take to get to where you're going as efficiently as you can. So in this case, the nodes would be locations on a map and the edges would be roads connecting them together. So why apply this approach for tracking data? I mean, at its essence, tracking data are relationships between pairs of points. You're taking points on a map and you're drawing lines between them. And so using a network analysis approach can help put a quantitative perspective on the kinds of things that we do just by hand when we're drawing lines between points. It can help you to find landscape scale patterns. So looking at habitat use by multiple species or multiple populations. It can help you to group sites based on their use by different individuals, and I'll talk a little bit more about that in my species example. And you can identify movement corridors that are particularly important for binding the network together. It can help kind of get beyond the static range maps that we see to incorporate this dynamic temporal element of individual movement. It can help delineate interconnected habitats and populations, which is really what we're talking about in today's session. Identify targets for monitoring, so locations or species that might be particularly important or particularly representative in a network context, and then to prioritize sites on a map for conservation action. So to get into the specific sea duck example, the interest of looking at sea duck movement and interconnectivity is that mainly the sea ducks are harvested species. So there's an interest in understanding how harvest is affecting different segments of the population. At the same time, they're very difficult to study and difficult to quantify because they breed in remote Arctic sites and they winter in the ocean. So they're never really in areas where they're particularly accessible or visible. Hunters over the past 30 years or so have been reporting that they're seeing fewer birds. They're seeing birds in, locations where there used to be large groups of birds used by fewer and fewer birds over time, but that's really difficult to quantify because there's not very good demographic estimates. And we have these really interesting migration patterns. So there's not really just a direct migration from a wintering site to a breeding site and vice versa. They use fall and spring staging sites that are often very separate and require separate routes. They're not along the migration routes. They actually require a detour to get to them. And during the fall molt period, they're flightless over a period of time when they molt their flight feathers. So these could be particularly important sites and periods of particular vulnerability that aren't really incorporated into that kind of binary breeding, non-breeding model that we're used to seeing. So some of the needs that my study was kind of trying to address or to see whether there are distinct populations that might be subject to differential harvest pressures, to assess potential risk factors across the annual cycle. So in all stages and all habitats, including those migratory staging locations that are distinct, and to prioritize habitat areas for conservation. So the data that I'm working with are part of the Atlantic and Great Lakes sea duck migration study, which is a large-scale effort that's been going on for almost 20 years at this point. I think the earliest data are from 2003. So this is one of these species that, as Autumn Lynn mentioned this morning, we're really getting into the realm of big data because we've been tracking them for such a long time. And the tracking data all come from implanted satellite transmitters. So the great thing about this study is that it was coordinated across the entire wintering range. So the sea ducks were captured on wintering sites at sea throughout their eastern range, from the Chesapeake Bay through the Gulf of St. Lawrence and in the Great Lakes as well. The project was managed by the Sea Duck Joint Venture and involves multiple partners in Canada and in the US. And so those colors on the map are 5 different species. The blue is black scoters, 89 individuals were tracked. The green is surf scoters, and the yellow is white-winged scoters, 83 individuals were tracked. The purple is common eider, and the red is long-tailed duck. So you can see the sample sizes are pretty huge compared to a lot of telemetry studies. And we have this multi-species kind of tracking effort across the same geographic range and basically the same time period, which is also pretty unusual and really a testament to the foresight of Sea Duck Joint Venture and the collaborators. So what I did was to start out with all of the tracking data and to separate out the resident locations where birds are actually present in habitat from locations that they're just flying through as migrants, because we're really interested in looking at sort of landscape specific locations. So the blue locations are identified by a state-based model as migratory, or when they're moving through an area quickly. And the yellow locations are identified as resident when they're spending a lot of time in the area. So it's kind of an initial step to get to the areas that they're actually using. So we assigned all of those resident locations to seasons, just depending on their position in the annual cycle, calculated habitat centroids to get rid of some of the variation in the data, and then used a cluster analysis to group those into sites on the landscape. So I can go into more detail on any of those steps potentially in the Q&A, but I'll go through them pretty quickly now to get to some of the results. So this is the global network that we came up with. So each of those is a cluster of sites that were used by individuals that are kind of separate from one another based on a cluster analysis. And so this is a view of the network where the size of the node actually indicates the number of birds that use that node. The size of the edge, so the width, indicates the number of birds that use that edge, so move between that pair of nodes. And this is a directed network, so the arrowheads are the direction of movement. And finally, the colors in the pie chart correspond to the seasons. So blue are wintering sites, green are spring staging sites, red are breeding sites, and yellow are the fall staging and molt sites. So you can already see this is starting to give you a perspective of not only where birds are occurring, but when and how concentrated they are and what routes they're using to get between those sites. If we break that down by season, you start to see some of the movements that are happening within the seasons. So obviously during the migrations, they're moving around quite a bit between sites, and you can see some of the sites that are the most important during the migration period based on the number of birds using them. But we also see quite a bit of movement within the winter. So even in that specific season, birds are using multiple different habitat areas. So this is an example of kind of what I was mentioning earlier about introducing this dynamic component into a range map. So this is for the black scoter. You can see their migration area in the typical range map as kind of this broadly defined habitat that can apply over both the spring and the fall migration. If you break that down into a network analysis, you can actually see that they're primarily using the Gulf of St. Lawrence during the spring migration and then using James Bay during the fall migration. So again, we're starting to get to a little bit more of a detailed understanding of where species are occurring and when, and how they're moving between sites. So doing that with all of the species, you start to see some of these patterns of multi-species kind of overlap in migratory routes. So black scoter and surf scoter have these pretty similar migrations, although surf scoter seem to be occurring more in the Gulf of St. Lawrence during the spring migration compared to during the fall migration compared to black scoter. And you can even see within the Gulf of St. Lawrence, there's a difference between the mouth of the St. Lawrence River versus the outer part of the Gulf that's used more during spring migration versus fall migration. And then with the white-winged scoter, you see a very different migratory pattern that tends to be more inland. And then with the other two species, the long-tailed duck and common eider, there's actually quite a bit of separation from those species. So common eider is not really surprising. They're mostly on the coast. Long-tailed duck are going much further north in their breeding range and skipping over some of those stopover areas that are really heavily used by the three scoter species. So getting into something that looks more like the typical migratory connectivity map that you can do with a network analysis is to group sites based on how modular they are, so how separate they are from the rest of the network. So in this case, a really strong connectivity would be similar to a high modularity, closer to 1, and a lower modularity, similar to weaker connectivity, would be closer to 0. Applying that to the sea duck network, we get a 0.15. So, and all individual species were also very low. So this seems to be a very highly connected or weakly migratory connectivity population. And there's not really a strong grouping of sites. Even with those Great Lakes birds that seem to be a little bit separate during winter, they're still interacting throughout the rest of the annual cycle. And then getting into some of the other metrics that network models are really useful for. So measures of centrality have to do with how important different locations on the map are. So the top two maps are in degree and out degree, which is how many connections are coming into a node and how many connections are leaving a node. So are nodes importing individuals or exporting individuals? And you see these nodes that are in red in the Gulf of St. Lawrence and sort of southern New England in both in degree and out degree are popping out pretty strongly as getting individuals from a lot of different nodes across the network, and same with the locations in James Bay. So these seem to be particularly important to the connectivity across the network. And those bottom two maps, so between us is the number of different paths through the network that the node is involved in. Again, the Gulf of St. Lawrence is coming out pretty high there, as well as southern New England. And finally, connectivity is if you drop a node out of the network, how many potential paths are lost based on the movements of individuals. So we get, different measures that give you slightly different information, but in this case, they're all pretty consistent in terms of those stopover sites and that particular wintering site in southern New England as being very important to connectivity across the entire network. Another interesting thing that you can do with network models is to look at representativeness. So often you might be interested in coming up with a species that's representative of a group of species, and especially in the case of sea ducks that are kind of all overlapping. There's definitely interest in identifying species that could be monitored to determine what's going on with the overall group of species as kind of an umbrella species. So looking at kind of how well each individual species represents the global amount of connectivity. You see that eiders are pretty terrible, which is not surprising given that they're a coastal bird and not using a lot of those inland habitats. But what's interesting is that long-tailed duck, which if you remember was also a very different pattern to the three scoters, shows up as being probably the highest level of representativeness, both for the global network and for the remaining species. So this is interesting because they're using different habitats, but also overlapping pretty strongly with the scoters. They come across as being a species that gives you probably the most insight into what's going on in the network as a whole. And then finally, you can start to break down what are the most relatively important sites for each species. So where are those species using the habitat more than the rest of the species to kind of get an idea of what may be driving differential trends in different species or different populations. So you can see, for example, the black scoters are particularly dependent on James Bay versus surf scoters tend to be using the Chesapeake Bay kind of more than the other species, and white-winged scoters tend to occur in the Great Lakes, probably more relative to other species, versus high Arctic sites for long-tailed ducks and coastal New England and Canada sites for common eiders. So you can also do something like this for sex. I won't show that in the interest of time, but if you're interested in looking at maybe what's affecting sex biases in populations or sex bias mortality in populations, you could look at what sites are relatively more important for one sex versus another. So what does this all mean? How is this all useful? So we really could identify some specific sites, and particularly these staging and molt sites that are kind of unique to sea ducks within their migrations that seem to be essential to population mixing and migratory connectivity. We see differences in node importance between species, but the part that I didn't show you is that they're all pretty similar for the sexes. So there aren't areas that are particularly more important for males versus females. But for certain species, they're using areas differently than one another. But there's little separation within at least the eastern portion of the range that we studied. There's not really any flyway specific separation. There's a lot of mixing happening, particularly on those staging and migration sites. And yeah, the long-tailed duck is also a particularly good umbrella species for the entire multi-assemblage in this case. So we're working on expanding this to the continental scale to also incorporate Pacific populations. And then once you kind of identified these key nodes, you can start looking at what are the potential threats, population drivers happening within those nodes, what are their habitat requirements when they're there, and how can that be used to kind of develop really concrete conservation strategies. So I'd like to thank all of my collaborators and funders. As I said, there's more people that are not listed on this slide than I could really mention, but there are a lot of people working on this project. So, thank you.
That's really fascinating, Juliet. Thanks so much. And thanks for taking a major leap, not just across taxa, but across silos and bringing in particle physics at the beginning of your talk. I pulled out my little handbook of Dr. Einstein's universe. So I tried to follow along as best I could. I don't have a question, but I wonder if there are any questions coming up in the chat. Anna?
Yes, we do have some for Juliet in the Q&A. So you spoke to this a little bit already, Juliet, but you said there are plans to maybe expand this to the West Coast and the Pacific Flyway. Are those plans concrete yet? Do you have sort of a plan?
Yes, I have the data.
Oh, awesome.
Yes, we're working on the models now. So yeah.
Cool. And the second part of that question in the chat is, going forward, do you think it would make sense to use Martin Wikelski's Icarus technology to look at the network across this vast area of the Pacific coast as compared with GPS tags?
Yes, I think that would be fantastic. Yeah, the satellite tags definitely have limitations, as other people have mentioned, and like they're not solar powered, so they have limited battery life. And yeah, there's, especially in earlier data, there's fairly high tag failure rates, but it's also pretty invasive for sea ducks. So yeah, developing technologies that can maybe give us more fine scale data and last a little bit longer would be great.
Awesome. Wow, there are a bunch of questions in the Q&A now. Okay, so another one for you is, if you used R for your analyses, what packages did you use for network analysis specifically? This person.
iGraph. Yeah, iGraph is excellent. I would highly recommend that. It's very straightforward to use, almost too straightforward.
Cool. Okay, Alan Harrington asks, I was curious if you could explain in a little more detail how you derive the representativeness metric and how this metric could tie into management.
Okay, yeah, so basically, once you have these measures of connectivity, each node in the network is associated with a certain amount of connectivity. So like you can drop one out and see how that affects the connectivity of the entire network. So basically what I did was just to take the species-specific networks, so the set of nodes that species are using, and look at, if you added together just the connectivity from those nodes, how much of the overall network connectivity would you represent? So that's a proportion of the overall.
Okay, cool. Another question is, I'm curious about whether you also estimated network level attributes. So I think you talked mostly about patch level attributes, and she's wondering if you looked at edge density, centralization, and what insight they might be able to provide about species resilience or sensitivity to disturbance.
Yes, I did look at them. I'm not sure about specific insights that they could give. I found that the sort of patch level metrics were kind of easier to interpret in terms of their importance. But yeah, with networks, there's kind of almost an unlimited amount of information that you can get from them. So narrowing it down to what's going to help you answer your question is important. So yeah, those overall network metrics are easy to obtain, but maybe a little bit more difficult to interpret, I would say.
Thanks. Tom, I don't know if we want to move into the general panel discussion.
Yeah, I just promoted this morning's speakers to be panelists, so you can you can all unveil yourselves and be present with your faces. Are we missing anybody? Let's see. I'm wondering if Martin is Martin available?
I don't see him.
Okay. So I've got Autumn Lynn and Stu and Kristen. So one of the goals of this portion of the workshop was to help people sort of figure out where, you know, where they might sort of begin the work on a particular species, and, you know, any insight that any of you might have in terms of what your breakthrough points were as you proceeded to work on individual species, whether it be wood thrush or whether it be nighthawk or lesser yellowlegs or the sea ducks, where your sort of enlightenment came in terms of how to proceed, and any sort of second thoughts you might have about how you would approach things differently using some of the technology or methodologies which have been introduced.
No.
Well, I can take a stab at that. So when we first started building our connectivity map for wood thrush, we were kind of just trying to get a good cover of the range, the breeding range of wood thrush and the wintering range, because we deployed on both ends. But now I think if I was going to restart this project, I would really take kind of the approach that Ellie took and really look at natural populations. You know, that Clark Rushing has a paper that created natural populations for wood thrush based on demographic data, and then really choose our locations based on that and filling in gaps across those populations. And so I think that's one thing that I would definitely do different, so that we can start asking these questions about differential population trends across the range. And then a secondary thing, you know, I was really interested in looking at timing of migration and carryover effects. And I thought that I would be able to do that with the GPS tags. But the reality of those tags is that, I mean, I mentioned this in my presentation, is you really don't get good temporal resolution. And so I think incorporating a mix of the two technologies might be really good for some species, depending on the questions you're asking. And especially since the light level geolocators are so much cheaper, depending on the geography of your species and the questions asked, I think they can work just as well as the GPS tags. Because I didn't really show this, but similar to Ellie, you know, I mean, different technologies, but we had tags that could get up to 50 points. But in reality, you don't actually get 50 points. On average, I think we were getting like 25 points across the annual cycle. And so you really have to be careful about what questions you're asking, given the fact that you have no idea when you're going to get those points, and you're not going to get as many as you think. Though for us, we got really good, we often got our locations scheduled during the stationary periods. It was really the migratory periods where we missed out. And I think that might have to do with the timing of the location estimates during stationary periods. We had the time set to be taken at night when birds were sleeping and still up in the canopy. Whereas during stopover, we had the locations timed for the daytime because we were interested in stopover locations, not migratory routes. But I guess those are some things I would consider.
If I understand Clark's methodology, and he'll speak tomorrow, his identification of natural populations is based on information coming from BBS, so sort of demographic information, correct? So, you know, how much time would it take, and what would be the difference if we estimated those natural populations using genetics? Or, you know, and maybe Kristen is the right person to ask that question to. I'm feeding off your idea, Callie, that wow, if we started with the natural populations, you know, that would kind of be interesting to sort of go from there. But
And I'm sure Kristen will say this, but those natural populations aren't the same thing as like genetic subpopulations. And I don't know, maybe Brant wants to jump in, but for wood thrush, I don't think it would work, but Brant, go for it.
No, I was just going to chime in to say that, that's one of the things that Clark and I talked a lot about when we generated that paper, was whether or not those populations actually reflect kind of true genetic populations. And I think the answer to that is probably no, but they're at least demographically, or based on the BBS data, demographically unified in terms of trend. So yeah, it would be interesting to try to overlay those two and try to come up with what are true populations, and then use that information to inform some of this connectivity work. So.
Yeah, I was gonna, let me just chime in. I was gonna ask a similar, I was thinking about that now after Kristen's talk. I was thinking about that for everybody's talk, is wondering how that really relates to the, the underlying genoscape. And I don't think Kristen's back on, but that would be really interesting to do, sort of a cross calibration across some of these techniques. And one of the things I remember from Kristen's talk and seeing some of her other work is you very often have a pretty homogeneous Eastern population that goes up, out into the boreal really, that's very distinct from some of these Western populations. So like with the nighthawks, for example, I mean, if you're finding what looks like weak connectivity across sites from the Great Lakes to the Atlantic coast, there you are, Kristen, so if you could correct me, that still might all be one, it's usually one genetic population across that area where you might not expect to see connectivity necessarily, unless there's just simply an east-west thing going on. Whereas making sure you're sampling some of these genetically really distinctive populations, that might be in the West, and then looking for the connectivity in relation to that distinctiveness. So that's kind of a different approach from just, a lot of these studies have sampled across the range. And then there is this question whether, even if they look like natural populations, demographically, whether those have an underlying genetic basis. I think that at some point connecting these will be really interesting.
So I can just pipe in there. Sorry, I couldn't figure out how to turn on my sound again. But yeah, so one of the things we're finding with the genetics is two things. There are these like situations where there's distinct splits, like we saw, like I presented with the willow flycatcher earlier. And then there's species where I would guess the wood thrush would fall into this category. Like I've been working with Pete on the American redstart, where previously with other markers, they saw just distinct, clusters that were maybe northeastern and then all of the boreal forest with other markers. But with the whole genome data, we can get really kind of fine scale isolation by distance and environmentally like signals of local adaptation that pull apart populations in ways that you previously couldn't have seen with other markers. So I guess and I agree, like the overlaying that kind of genoscape, a really fine scale genoscape with like the Clark and Brant method of demographically defined populations is something I've been really trying to get a grad student interested in. Like this would be such a cool project. So far, nobody's taken the bait, but if you want to work with me on that, let's do it. I think it would be so cool.
Well, we can try and tackle it for nighthawks eventually, Kristen, when we get all the blood.
Excellent,
If anyone
excellent.
out there wants to learn how to blood sample nighthawks, send me an email.
Can you do your technique with feathers from museum specimens, Chris?
We can, yeah. It's sensitive because it's destructive sampling. So, but we've certainly used that as a method to fill in gaps. And then there's another student in my lab who's also been, linking back to the local adaptation question, is using the museum specimens to look at changes in these environmentally selected loci over the time period of climate change. So yeah, there's all sorts of ways that museum specimens can augment the genetic studies.
Yeah, because there was a question in the Q&A about how dependent it is on catching birds, you know, if there's actually less and less banding going on. And again, I was thinking with the nighthawks that there are so many named subspecies that are really distinctive, but where we might not be able to go and do the current sampling. That would be a really interesting, if nighthawk is our model species.
Yeah, yeah. I mean, less and less banding. Do you think our people are still catching birds though? I mean, will people still need to catch birds for a variety of these techniques? So
They do, but I would just add to that and say that museums in general are just this gigantic storehouse of information that could be added, whether it's for genetics or morphology, that we still really have to tap a lot more.
So what happens when you apply what Julia was describing, the network analysis to a GeneScape model? I mean, how do you what would
excellent question, Tom.
what would we say? Oh my God, or wow, or unbelievable?
Well, I hope so. And, you know, our paper just came out in conservation biology this week. So you can go and read this. So we did it with Kaz Taylor and Jim Sirocco and Ryan Harrigan. We just tried to demonstrate how you could use GeneScapes as the foundation for network models. And so I was very interested to listen to, you know, the network talks in this symposium to get, you know, more ideas. I think, you know, certainly as a foundation for defining breeding nodes, it's really straightforward. The thing I struggle with the networks is how to define the non-breeding nodes. Like, and I would love like a panel to come up with that. Like, do we just arbitrarily carve up nodes? Do we do it by geographic boundaries? Or is there some way of doing it that's more informed by some of the migration data that you guys are all collecting?
Yeah, I would chime in on that too.
Okay, and say, great. Yeah.
Well, I mean, I think that the defining the nodes part is really like the hardest part of developing a network. Like once you have the nodes, it's easy to get all the information, but like having the nodes structured in a way that's meaningful, I think is like the key part of the network model. And I don't know that like mine was just based on spatial clustering, but like some of the clusters are pretty large. Some of them are like, yeah, you can tell that that's a defined area and it makes sense. Some of them don't necessarily make as much sense. So I'd almost argue, like if I went back and did the analysis again for choosing nodes based on like something that can be conserved, like a conservation unit almost, yeah, and using that, because I think in terms of utility, like then you can go and you can say, okay, this particular refuge is, you know, contributing X% of the connectivity, therefore it's a priority. And then like, yeah, you know, managers can use that versus just like, oh, it's kind of this big area that has a bunch of points in it. So, I'm excited to read your paper.
Yes, okay, I'm glad to hear as the real network expert on the panel, you agree. So that's exactly what we did in this conservation biology paper, is tried to say like, hey, it makes perfect sense to define the breeding nodes by conservation units that are defined by the genetics, because then that really sort of sharpens, in my opinion, read the paper, you can argue with the paper, but I think it sharpens our idea of where then you would prioritize those you know, conservation units that are more in threat over ones that are not as in threat on, you know, throughout the full annual cycle. And so that's what we tried to do, is use that approach and then show how that really helps you kind of hone where to focus prioritization. But we still struggle with how to define winter or non-breeding nodes. I don't know. Yeah, I don't know if we did. I don't think we did. The reviewers didn't like what we did, but we tried our best.
So. Autumn, Lynn, do you have any thoughts on that?
Not too many, except it makes me recollect historical fisheries literature, that I think this question has been kind of bouncing around in fisheries for a long time. And thinking about the idea of stocks and conservation units. And these are units that are managed, and they have some relationship with genetics or stream origin. Or, one example that comes to mind that my husband actually worked on was studying Chinook salmon. And there's an endangered stock and multiple non-endangered stocks that when they leave their freshwater environment and go to the ocean, all of them are fished. And so trying to set harvest limits for the endangered stock while also protecting the fisheries for the non-endangered stock when they're all mixing out there in the ocean. And I think there's just this comfortability with focusing on conservation units and stocks as kind of an endpoint and as a goal of a study. And that may sometimes be separate than what we might be comfortable with ecologically or from life history or genetics. So I think if you have that mind that you really want to inform management, talking first with the people that will use the results of your study to find out what scale and what unit and what region and focal geography is important to them, I think is really important. And that drove some of my own work studying marine migrations, that asking simpler questions than I would have from a scientific viewpoint. And maybe on geographies, I might not have separated out migratory species into, but because that's what the managers want, that's the unit that they would manage at. So reframing some of our analyses that they're done at the same unit of management, I think is really important.
I would just say, like, yay, I completely agree. Like, a shout out to the fisheries management community. It's probably not coincidental that I'm married to somebody that works for the National Marine Fisheries Service. And so everything we've done with the Bird Genoscape project has been, it's like me trying to be like, look, fisheries biologists have been doing this for centuries. This is the way we can manage birds. Like, let's do it. We can use a lot of the tools they've already developed, and that's what, that's what we're trying to do. So
I'm getting a key message here. Start with fish and then move up into the air. Is that what we're saying?
I think the fish people started a long time ago. Yeah. Unfortunately, they're working on a commercial, commercially viable set of species. And so they're much, much better funded than we are, which is another source of strife between my husband and I. I'm just like, it's so unfair. You have like the entire federal government supporting you.
Yeah, coming from
for NOAA, so similar.
Coming from non-game seabirds to sea ducks, I think I agree that there's like harvested species, there's a certain perspective that doesn't exist in non-harvested species that could be really informative for conservation in terms of how we think about populations.
So another question that comes to my mind, and then at some point we'll go to the participant of the webinar and get questions from the field. And I'm thinking about using the geolocators that have to be you have to get them again to download the data, and the sort of, especially the light level geolocators with their sort of fuzziness in terms of how you determine the exact locations, particularly latitude. So what would it take to substitute for species like wood thrush that I think are too small to carry the Icarus type devices? When do you move to, or what would it take to have MODIS tags for those species, and what sort of a network would be required to get the same kind of information that you are getting with the geolocators, the archival units? And how close are we to that? And what should our investment strategy be in terms of both receiver deployment, and where do we put these receivers? That kind of question. Stu, I guess that's really up to you.
Yeah, and my kids just came home, so forgive any background noise. I no control. I think a lot of the resolution is already there. It's just a matter of figuring out how to model and interpret the data to get what we want out of it. One of the points I was trying to make in my presentation is that there's so much there that we can start to tease it out. And then in terms of how we distribute the network. That really depends on the species and the questions and the targets and the priorities. It's an easy question, but a lot of that is happening naturally through the strategies that are being deployed in different working groups by creating strategic fences or grids. And we haven't been very selective. We're sort of going on the just blanket the hemisphere approach because every state has its unique interests, and every region has its unique interests, and grassland birds are doing different things than shorebirds. So you kind of need to cover everything, and that's quite a task. But in the meantime, movement modeling and some advances in that field can help fill in the gaps, and that itself is a huge gap in what we can do with that type of data. Figuring out what we can get out of all the holes. I don't know if that addressed what you were looking for.
Kind of, and we'll touch on this a little bit tomorrow when we start talking about some of these other methods for getting life history data or vital rates data, like maps and MOSI stations. And so my question then tomorrow would be, what do we get an order of magnitude boost in power of inference if we pair a network of MODIS stations with a MOSI station or with a map station?
Yeah, so I was wondering, let me just jump on that also, Tom. Are there any examples? I mean, the MODIS tags are so small. Are there any examples of a bird that carried a geolocator and a MODIS tag, let's say, so that you could start to calibrate across the methods? Because if there are questions and imprecision about the light level, you have to model where you think that bird was. But if you got an actual ping, you could, theoretically calibrate those. I don't know whether any of that's been done yet.
We've played around with homing pigeons and GPS tags and MODIS tags, and we discovered it's noisy. But I think we're getting to the point with the miniaturization of both technologies that we could start to do that, particularly with archival or pinpoint tags and Icarus when it comes online. Even some of the early generation direct satellite tags could do it now. Or we just choose, like, stop being so picky and just do that kind of an experiment on a duck and just load them up with, as much as they can take to discover those questions.
I think that can also be done with, we've done it with isotopes and tracking devices on the same individual birds. And you'd be surprised at the variation that you get in both measures. So one doesn't necessarily validate the other, unfortunately. And I don't know, Kristen, have you done it with genoscapes and tags, archival tag?
Have I done what? I was trying to respond to the chat.
Taking a genoscape approach with, assign birds back using a genoscape that you also had tagged with an archival device or another sort of a tag, tracking tag.
No, I mean the first. Well, so we have some data with the painted bunting, a collaboration with Andrea Cantina at University of Denver, where I believe he has GPS data for some of the individuals we have genetic data for, but we haven't actually directly compared them. And then the other one where I'm excited about is the Robin collaboration with you and your group.
I think a double tagging experiment, that's what we called it back in the day with albatross data sets that had either a geolocator and an Argos satellite tag or an Argos satellite tag and GPS tag. A lot of the larger tags collect Argos and GPS simultaneously. So you can actually use those data to help model and refine the location error estimates for previous tracks recorded in the 90s when you really didn't have GPS data to compare. So those double tagging experiments were used to really ground truth and make better utility of the data sets collected in the 90s for geolocator albatrosses and then Argos albatrosses. And I think a double tagging experiment with nanotags and a small GPS tag would be really, really interesting to help also refine some of the modeling approaches for processing nanotag data.
It's not as particular or individual based, but we do currently have quite a lot of data of the same populations, however you want to define them, particularly for knots, where you'll have knots with a lot of geolocator data and we'll have nanotag data from the same time and spaces, Bahia Lomas or Delaware Bay. So it's not the same individual, but it's the same, population, however you want to define it. That may or may not provide some insight. I'm not sure how, but.
So I have another question. This is a tough question. So going back to the whole purpose of these workshops, which is to try and figure out the causes of decline for species, particularly species on the brink, or species that are losing a major portion of their population numbers, and recognizing that there's a certain degree of urgency connected to making those decisions, and we care about understanding what are the immediate causes of decline because of the ability, therefore, then to target conservation action. So it's not just an academic exercise. We want to know so we can do something about it. So just with the information that was presented today, where we're really mostly talking about migratory connectivity, there'd been a few statements and different folks talk about, feeling pretty confident about maybe where in the life cycle limitation might be occurring. Do you feel, and I think this is a good question because of the urgency issue, we can continue to gather infinite amounts of information on every species, or we can say we've got enough information now to really target conservation action. So do you feel with any of the species you've worked with or about, we're at that point, just understanding the migratory connectivity of the species?
I'll tackle that one, Tom. I think I certainly don't feel that way for nighthawks. I think just because we don't really understand what those connectivity values mean for demographic effects yet. I mean, we can kind of theoretically understand what a 1 means and what a 0 means, but what does the 0.3 mean? What does the 0.7 mean? What does the 0.15 mean? And this goes back to the question that Anna asked at the end of my session. So I don't know that we could sort of jump right to conservation action, but I think the idea is that the connectivity at least pinpoints us in the direction of, okay, well, let's try these ones first. And let's relate those to vital rates the way that the folks are going to tell us to tomorrow. And if we keep going down that track, then yes, we'll get to conservation action. If that doesn't work out, then let's come back to connectivity. So I think that road to recovery is not always unidirectional. I think there might be some 2 steps forward, 1 step back as we work down it.
Yeah, we have talked about putting roundabouts in the road because for that exact reason. So.
I think for wood thrush, oh, sorry, someone else going.
No, you're good.
I think for wood thrush,
no, go ahead.
Pretty good understanding of migratory connectivity. We're still missing some populations, especially in the south. And like I showed how, we're having basically the global population funnel into the US in one particular region of the Gulf Coast. So you can really isolate one area that all wood thrush are probably using in the spring. But in terms of where it's best to deploy very limited conservation funds, I think that's where you really need these population models to assess, we're going to get the best bang for our bucks by investing here for this population or here for this population. And for wood thrush, there have been population models developed using connectivity data, and they've kind of said both that wood thrush are probably limited on both the breeding and wintering grounds, so we still need to figure out for individual populations where it would be best to spend money. Brent or Pete might have a better.
I mean, I could say for willow flycatcher, I think we have a really good sense of migratory connectivity, and actually several of most of the GeneScape species that we've completed GeneScapes for, we have a good estimate of migratory connectivity. But I think that in terms of being able to identify the specific drivers, the title of one of the workshops was, the smoking gun. I think the problem is that there isn't a smoking gun. I think climate change is a disease that's affecting largely every taxa. And then you overlay other anthropogenic stressors on top of that, and you already have kind of in a sense, like immunocompromised entire, species complexes that then, are being driven further into decline. That's probably biased by how much time and energy my lab focuses on climate change, but I would say it's hard to escape the potential, the current and future impacts of that across the full annual cycle, unfortunately. I'm such a Debbie Downer, sorry about that. But.
Well, Christian, let me ask you, because that is, and it might be ultimately true, and that's kind of the alternative to this species by species smoking gun type approach. But the fact that there are some species that are doing really well in the same environments, or a species is doing well in part of its range and not its other. So it's like this, mystery novel kind of digging in and getting from that broad scale threat to what's actually causing it here versus there. I don't know. I think that there's enough, if everything was, and some people think that's what our paper showed, and it isn't, but if everything was declining, then yes. But the fact that there's so much variation means that there has to be, I think, some of these more smoking gun type nuggets going on, under the umbrella of your disease.
Can I ask a question related to that? I don't know if I'm remembering this right, but Kristen, when you were talking about the different adaptability to climate change and different climatic niches, that was within species, right? Across different populations. And I'm wondering if you've looked at that at all, across species, and if climate change is one of the big drivers, if species that are doing okay are ones that are more flexible climatically.
Yeah, that's exactly what we're finding. So I sort of alluded to that with the willow flycatcher example, and also looking at climate specificity of the different locally adapted populations across the full annual cycle. And the ones that we are finding are doing the worst are the ones that are, our genome scan data suggests they're most strongly locally adapted on the breeding grounds, and then they're following that climate niche. And those are the ones that are already on the endangered species list. Whereas ones that are able to switch and be more adaptable. So maybe they're I can't remember who it was it what somebody was talking about the wood thrush and being but yeah, the ones that are able to switch across between different climate niches are doing better. And then when we look at the species level, what we see is that it seems like ones that are generally more adaptable and able to shift more quickly are doing better, overall. But yeah, so I think that's definitely something going on there. And I don't know, yeah, it would be good to quantify that in a much broader context across both species and within species.
Just to clarify one point, we aren't thinking that this is a smoking gun effort. It's the plural, smoking guns. It's definitely a multiple stressor sort of a thing, but there may be, and that's always been the case. But lots of people are sort of thinking we're thinking one limiting factor, but it's varying in space and time by all means. Identifying this as climate is really challenging, and very few studies have actually identified climate itself as a limiting factor in the past. Probably be something really important in the future, but that doesn't mean there may not be one factor that explains most of the variance that, if treated or dealt with, could really, like harvesting in Laura's case of yellowlegs moving through Guadeloupe, that would explain a lot of the variation in mortality. But that doesn't mean there isn't something else that's going to rear its ugly head that could be equally important for other populations.
So I think all of this, I was going to say, I think all of this discussion relies on the fact that we have good knowledge of demographic trends, which for a lot of species we don't, especially a lot of species that I study, seabirds. But I was chatting with Laura about this too. What are the differential population trends for lesser yellowlegs? And there aren't good data for that. And Ken and I have talked about this a lot too. But I think for some species, back to your question, Tom, we actually know more about the migratory connectivity than we do about the demographic trends, which is just crazy to me at this point.
Well, tomorrow will be an opportunity to dig more into those demographic trends and the vital rates and the relationship between getting those in the context of linked populations. So that should be pretty exciting. You're all pointing to that discussion tomorrow. I did want to take Kristen's suggestion though and recognize the fact that my advice to golden-winged warblers follows precisely from what you said, which is share genes with the blue-winged warbler and you'll be in better shape.
I just want to make a couple of, I know we're approaching the end of the hour, but I'm seeing some questions coming in, that we're obviously not going to get to. And a couple of them are, really hard questions that I just want to acknowledge that because we are still talking up at a pretty high level and in some cases even a conceptual level. And so one question, for example, from Ted Chesky is, with the red knot, even when you do know exactly what the smoking gun is and where it is, then it's still such a complex issue. That doesn't ensure the recovery of the species. And obviously that's true. And that's where we're heading in the future with this work is to, talk more about implementation and what it's going to take to actually recover once we do have the information. And then the other question was about, we're talking at these large scale, range-wide, continental levels, and people really want to know what to do locally. And so stepping it down, that's another whole sort of set of next steps, I think, once we have a lot of this information, is how does it apply in a specific area. So I guess I just want to acknowledge that, people are asking those kinds of questions and they are really hard to answer right now, but, we're not ignoring those. That's really where we're hoping to go in the future. And right now we're just really trying to, kind of discuss how to get there and how to get a lot of these specifics.
And we do have some time left. As long as the panel is willing to stick around for a few more minutes, let's say 10 minutes, to go to the Q&A, Anna, and see what might come out of that for good questions for the panel to mull over.
Yeah, well, like Ken said, some of these questions are pretty big and tough to answer, but I'll try a couple of them. Okay, so Andrew Cox asks, oh no, people keep asking questions, so they keep moving. A lot of the really excellent work presented today is occurring at large spatial scales. How do we step this work down to smaller spatial scales to help managing entities such as states and NGOs prioritize their work and implement the on-the-ground management needed to restore these bird populations?
Yeah, that was one of the ones that I had grabbed. I was sort of thinking ahead that the stepping down and the kind of question Sarah Kendrick asked this morning as well is, even for the connectivity stuff, you're talking about the nodes and talking about, whether they should be practical based versus biological based. And I was thinking about that question from earlier about the states. I mean, every state wants to know where their birds go and what their connection is and how much contribution they can make, to a species. So that is another whole way of looking at it that maybe sometimes we're not thinking about.
Okay, John Alexander says, with this discussion turning towards demographic trends, I'm interested in Stu and others' thoughts regarding using MODIS to help tease out what we can learn through constant effort misnetting methods, which, yeah, will be talked about a lot tomorrow.
Yeah, I'd be interested in what others think about this as well, but there's been some only high-level talks about how we can combine the methods of whether MAPS and MOSI or different forms of migration monitoring with MODIS that allows us to drill down. So just off the top of my head, we have a whole range of historical stopover data from all these constant effort misnetting sites, but a little bit of application of real-world MODIS data could help to quantify or confirm modeling of stopover data. Survival is probably the biggest one, where we can actually test true survival versus modeled survival from constant effort data, as well as dispersal, probabilities of recapture, and all those ideas, how individuals are using the landscape and how a constant effort site may or may not represent what's actually happening. So off the top, those are a few good ideas. Each one needs to be flushed out a little bit more. So John and others happy to chat about that at any time.
All right, there are a couple other questions in the Q&A. Species by species research to inch up the pyramid is quite slow, particularly in parts of the world where there might be less funding to advance research quickly, yet declines continue apace. And this is a little bit what Tom asked earlier. Do we know enough to consider no regret measures for conservation that can help multiple species while we continue to fill knowledge gaps? Yeah, so that kind of gets to this question again of like, how much do we need to know before we can take action?
Yeah, I can address that. So we aren't at all proposing that we shouldn't be continuing to do broad scale conservation to reduce known threats like habitat loss and really critical areas. That has to continue. And that's what we've been doing for the last 30, 40 years with groups like TNC and Conservation International and other groups, lots of land conservation trusts. That does need to continue. But what we're saying is that there's this group of species that are getting precariously low in their population sizes, where we need to sort of now think about that and getting the information necessary to do that protection. But by all means, we've got to do simultaneous things. We've got to multitask. Unfortunately, our job has just gotten a lot harder. So we need to continue those other efforts, probably ramp them up to identify other, the whole 30 by 30 effort is hopefully going to be a priority of the Biden administration, in addition to climate change efforts, where we're really trying to save 30% of the Earth by 2030. That is a huge effort, and that's broad scale conservation that will protect multiple species. And my hope is that that's not just in North America, but that extends down into Central America and the Caribbean and South America. That's a big ask, but my hope is that's what we do. So again, I think we have to multitask in our conservation moving forward. And some of that is species by species, some of that's broad scale conservation.
And another aspect of that is, as we, it's an iterative process. So another piece that I think we've not been very good at is evaluating what's working and what isn't working, and feeding that back, into the model itself. And there are so many programs out there that don't even have monitoring associated with them. So we're trying all kinds of things, and we don't know which of them might actually be helping the populations or not. So I think we need to do a better job of connecting the broad scale on the ground work, but thinking of it as an experimental phase that can feed back into these, into these smoke and gun type models in a more iterative fashion. And I think that's another aspect of multitasking.
Hearing a pause. Oh, wait a minute.
I was just reading one of these long questions, but it's a good one. Katie Leonard asks, there are so many aspiring biologists ranging in ages who may have valuable input on different solutions or steps to be taken, potentially adding a new take on things. However, they often don't know how to get their ideas heard or across. How do we get these small or new ideas to be heard by the larger scientific community to continue to inspire and create the possibility to make large scale differences?
You take that one, Anna, since you're an example.
Yeah, I mean, I think that workshops like this are super important to be able to hear from a lot of different experts and from people of all ages. I mean, there are a ton of grad students here speaking to just kind of get an idea of what the cutting edge research is, where people are thinking we need to go with this, so that, yeah, students and young people who are thinking about these things and want to be able to contribute can sort of see what the research needs are, and get an idea from the experts of what the research needs are, and also just have a chance to connect and chat with people in the chat, or be able to figure out people to email who are kind of echoing some ideas that they might be thinking about. Yeah, so I guess I would encourage anyone who is listening, who is an undergrad or an early grad student thinking about ways to plug into this kind of research, to reach out to any of the panelists here. And there is going to be an hour at the end of the workshop on Thursday that's totally devoted to questions from the audience. So definitely, if you have thoughts or ideas or want to figure out how to get involved in this research, use that to connect with people and get your ideas out there and heard.
And also, once again, we'll keep the chat open, and the Q&A in the chat will save both of those instruments, and there'll be a record to that. And if you've registered for the conference, we'll make sure that all gets distributed to you. So, and we'll try to do as much as we can with that in terms of reports and disseminating that information. So your ideas recorded in the chat will be there forever. Sort of. Anyway, I want to respect our panelists' time now, and I'll give that extra 20 minutes or so for the chat to be open for people to just kind of do their thing and write and toss off ideas. And I think the order in which we blink out will perhaps indicate, oh, gee, what's happening here? Where the hope for the future lies. Oh, there goes another old white guy. Oh, whoa, what's going to happen here? Oh my gosh. Thanks everyone for, oh, wait a minute, I see one more. Thanks everyone for participating in today's workshop, and we look forward to seeing you tomorrow morning. Thanks again for all the panelists and presenters. It's been great. And once again, see you tomorrow, I hope. Bye-bye.
Thank you all, even though Tom got rid of me.
Thanks, everybody.
Tom or Anna, did you guys see my, well, Anna saw it, but I've started moving people over to panelists for tomorrow, but we'll have to figure it out.
Yeah, I think that makes a lot of sense what you figured out.